So, in the United States, it's become big to show disclosures. I don't know if you guys follow Memorial Sloan Kettering in the newspaper. So, we always had to report conflicts of interest, but even more so now because it's under scrutiny. That said, it's completely irrelevant to this talk, and I just figured I would make a political comment given my political sort of acumen. So I wish I worked on osteosarcoma 30 years ago. 30 years ago, when you started an osteosarcoma, there was like 9 or 10 drugs out there. And what the oncologists were able to do is to combine every possible drug in every possible combination to basically yield a relatively small subset that worked, and then figure out how to sort of combine them together. And basically, that empiric approach of trying everything yielded a lot of progress. And obviously, this is showing the MIAS study, which we're all familiar with, the randomized study of chemo versus no chemo. So clearly, chemo works in this disease. That said, there's a million ways to show this. And actually, when I sort of review grants, I get annoyed by this part of the grant because I take it personally. But if we look at the last 30 years, there's really not been a lot of progress in osteosarcoma. It doesn't matter how we show this, whether it's SEER data or comparison of outcomes across clinical trials. Enough said on this topic. What the Children's Oncology Group published was really an editorial and just a statement from each of its disease committees of how it was going to go forward. And in 2013, they published the blueprint for research in bone tumors. And one of the statements they made very clearly is that there would be no phase 3 trials from that point forward, or at least, in the immediate future. And what you have to recognize is even before the URAMO study in North America, we had a study led by Paul Myers, which everybody knows about because of MTPPE. But basically in North America, it did not change the standard of care. What there was a recognition is that the NCI is resource constrained, and these large trials weren't going to be possible in the absence of really compelling data that there's going to be a positive trial, meaning how many big negative phase 3 trials are we going to do? And again, a lot of that was about cytotoxic chemotherapy, not about newer agents, but we didn't know which newer agents had activity. So that led to sort of the next set of slides. So the question is, if we were going to screen drugs very quickly in the phase 2 setting in patients with relapsed osteosarcoma, what was the best approach to do it? And, some of the idea around it is maybe we need to do a randomized study, even in the relapse setting. And then the question really became, is that appropriate? And so what the Children's Oncology Group did is it aggregated the results of its phase 2 trials that had been performed in osteosarcoma previously. I know there's a pointer, but I like using my finger. So on your left, what you're going to see is 7 trials that were 7 different agents, all in patients with bulk disease. On the right, you're starting to see sub-analyses. And basically, what you can see is these results are remarkably consistent and unfortunately remarkably poor. And what that immediately drove is a sense that we can't randomize patients to any sort of standard of care because the standard of care has such a dismal outcome that we should go ahead and use a historical control. And so if you look at this aggregated curve, the 4-month PFS was 12%. Which is sort of a low number and was published in JCO and remarkably consistent. And what we sort of said as a philosophy is we're looking for agents with enough activity that we can use these agents to pull forward to justify a phase 3 trial. So there was a lot of possible bar setting, but what we did is took the upper limit of the confidence interval, which was 19%, and just doubled it. We can sort of talk about the statistics that went behind this, but frankly, that's exactly what we did. And because Americans don't know math, we rounded to 40, so it wasn't 38. So basically, that was our sort of bar. And then the question became what agents? And, my talks, I always move back and forth between biology and clinic, and so we're sort of shifting to the biology side of things. One of the things that had been immensely successful within the children's oncology group was actually its tissue banking effort. So the tissue banking in osteosarcoma dated back to the first study started in 1998. When this hit 2,000 specimens, I stopped updating the slide, but basically we're about 2,600 biopsy specimens at this point. Not all of them are frozen, but there is an abundance of specimens. So this was really, really successful. One of the things that also came out at about the same time is some of these specimens were used to derive patient-derived xenografts. And going back to a conference that dated back to the early 2000s, there was discussions of who had what models. And basically, the existence of all these models led to the creation of an NCI-funded consortium to screen in a blinded manner drugs through the preclinical testing program. So basically, there was a series of laboratories funded to sort of take drugs as a pipeline with the goal of doing something between 8 and 12 drugs a year and basically screen it through these particular model systems. This project, I think, is kind of interesting because, I started at the beginning by making fun of empiricism, but what this kind of allowed is in the mouse to go back to empiricism and try every drug that was out there and once again just go to a sort of a screening approach and let's just do what we're going to do. So through this sort of consortium, at this point, it's been in existence for about 13 years. We've screened over 100 drugs. A lot of them get published in Pediatric Blood and Cancer just because there's an agreement so that there's high turnover of data. Not that we think that's the best journal in the world. And again, what you're sort of seeing is in the red and the blue is most of the drugs weren't that active. But the red and the blue were the things that were the hits, and particularly among the microtubule inhibitors, which would have been red, but I made them blue because those drove actually COG phase 2 trials. So when we look at a single example of the data, I think there's a couple of points I want to make. These are what the papers look like. They always come up with a heat map, which gives you the level of activity in the PDX models. This was considered a very positive model. Hence, this went to a phase 2 trial after validation that the tumors express GPNMB, the target of this antibody drug conjugate. But even when drugs are active, it's about 3 out of 6 that respond. We think that's pretty good. Some things none respond, but to sort of make the sense that this is a uniform response, it's just not there. Again, I wanted to highlight that this isn't the only way we selected drugs, and this is sort of highlighting Rama Koka's work, who's up in Toronto. She had developed a transgenic mouse that sort of suggested rank ligand may be relevant in osteosarcoma. So this particular transgenic sort of suggested denosumab may be an effective therapy. So we figured we would try that as well. Again, see, we're back to empiricism, and we're going to screen a lot of drugs in the phase 2 setting. Again, osteosarcoma, at the end of this, I'll get to the hopeful part, but it's always a long time sort of being, disappointment after disappointment. I'm kind of sorry about that, but that's kind of the way the talks these go. So the red line through each of those 4 agents is because these have all been reported in one place or another, but basically all 4 of those agents did not meet the threshold. We could argue that some of them had activity, but none of them made the 40% bar. And certainly, our initial goal isn't to lower the bar to make something pass. See the prior concern that in the phase 3 trial, this once again may not show a difference. We have other things in the pipeline. We have a sort of protocol looking at radiation and immune checkpoint inhibition and, this vaccine approach. But basically, this was a bit disappointing. So we have to go a little bit further into the science, and I sort of am using a slide I got from Alejandro Sweet Cordero rather than something from his publication, which came out in 2018. And basically, the point of this paper, like many others who have profiled osteosarcoma, there are a lot of copy number alterations in osteosarcoma. And I think what he started to do, which is different from other groups, is, if we look at our brain tumor colleagues, they have no issue with dividing their tumors into 8 groups or 16 groups or 32. And every time I stand up at a meeting that's mixed and I say, how many forms of medulloblastoma are there? They tell me I'm wrong because it's doubled again. So they have no trouble saying it's more than one disease. If you start looking at this, there seems to be different categories of drivers, accepting that there's messiness, that some have more than one, and it's a little bit more complicated, but there are ways to start grouping them. The other thing he started showing in this paper is he started having a relatively small collection of PDX models, and he showed that the PDX models, by and large, also can be basketed into the groups that the samples represent. Again, the concept of osteosarcoma being more than one disease, seems to be something that I have to try to prove to folks. But at the same time, there are so many differences between patient to patient. And this just shows you histologic subtype. And I'm going to show I'm not a pathologist because all the slides are blue and like washed out colors. But you got the point, meaning the histologic subtype of osteosarcoma, they all look very different. We don't care. We treat them the same. The outcome's the same. There we have enough data to support that, but in the molecular bucketing, maybe we don't. When we look at the PPTC models, we see the same thing. So the paper that came out from Alejandro Sweet Cordero's lab is published. The data publishing the sequencing on all of the PPTC models has not been published yet. That manuscript's in preparation. What this is going to contain is about 250 different PDX models made by the different labs. In the osteosarcoma space, there's like 30 plus models. That will be part of the manuscript. There are categories of the human tumor that he wasn't able to see in the PDX models, namely the IGF-1R amplified. Those in a larger panel you start seeing. And it sort of immediately leads to the question of when you did this 100 drug testing through the PPTC models, which PDX models did you test? And the answer to this moment, even through this moment, because we haven't sorted out the data, is I don't know. So it's entirely possible out of the 5 or 6 different categories of osteosarcoma, we only treated 2 of the types, and on the trial, phase 2 trial, it wasn't the type that responded. So I think we have to think about, molecularly guiding some of our decision making. One of the things he did in his sort of paper is sort of broke them down by groups, largely driven by what are the drug targets of each of the groups. And when you start looking at the drug targets, there's not great drugs matching to each of these, but he was able to show that there's a higher level of activity in the tumor samples in the PDX models that have the derangement that would be predicted to produce sensitivity as compared to the others. And there's a lot of statistical analysis because it's a little bit leaky, but it sort of helps in the selection. When we start talking about precision medicine, then the question really becomes, is precision medicine really need to do osteosarcoma trials? We've heard enough times, in enough different meetings, the association of track alterations and activity with LOXO 101. That is histology independent. So there are going to end up being osteosarcoma patients on precision medicine trials. And what you're seeing here is this Children's Oncology group has a match study, which is sort of a molecularly derived treatment protocol with multiple sub-aims that keep growing with different drug treatments. And this may be a way of beginning to develop getting a sense of activity. That said, there are folks who believe we need to do this specifically in osteosarcoma, particularly driven by an advocate, Teresa Beach, who's been prominent in North American meetings. It's a person who lost her son Daniel to osteosarcoma. But she was actually tenacious and got Foundation 1 data and started using Foundation 1 data to start bucketing patients into different groups. and is sort of driving a little bit of an initiative to try to sort of have this happen in an osteosarcoma-specific manner. So, the title of the talk I was given was sort of lessons learned. So, I have to sort of go back to what are the lessons learned. And I also have to remember that, idea after idea has sort of failed in osteosarcoma. So let's sort of think about how things could possibly go wrong. So one is we just don't know enough. When we look at sort of profiling osteosarcoma, we see it's a copy number gain and loss disease. Most of the targeted therapies are sort of targeted to mutations that activate or inactivate. Our drug pairings aren't particularly good. So even if we have a good molecular subgroup of osteosarcoma, if the drug match isn't good enough, maybe we won't see activity, maybe we will. Maybe we should sort of ignore sort of the genomic features and start doing proteomics, epigenomics. Maybe that's going to yield better targets than what we have thus far. We already know we've been fooled by heterogeneity, and I would say we've been fooled by the heterogeneity of the disease overall, but how sure are we that even within a given patient that there isn't heterogeneity within their tumor? So when you sample a particular part of a tumor, are we really sure when you go to another site of the same tumor that it's entirely going to have the same molecular profile? There hasn't really been enough samples profiled from different locations to begin to characterize whether that may or may not be the case. And we're kind of in the infancy of doing this. Lesson 2, if we're going to talk about tissue banking, is getting the right tumor samples. So I think the COG tissue banking effort was phenomenally successful. It was a wonderful effort, 2,500 samples. All 2,500 samples just about are newly diagnosed biopsy samples. Because back when we collected them, the idea was we were identifying biological prognostic factors. So you needed upfront samples and you needed clinical annotation, and that seemed to be all we needed. I think if I was going to do this over, I would think much more comprehensively about this, meaning I actually think, if we're trying to treat patients who have refractory disease, maybe the relapse samples were probably more important than the biopsy ones, and we didn't collect those. We didn't collect the definitive surgeries. We didn't collect multiple time points. And so a major flaw in our thinking was not only the type of tissue we collected, but the time points we collected it at. Obviously we can't fault not getting Streck tubes and things like that that preserve DNA because they didn't exist back then, but we could have thought of this. Lesson 3, I think, is critical to advancing the field as appropriate study designs. This is sort of a very short sort of statistics, experiment where, if we look at the Uramos sort of schema where there's a stratification after histologic response, you require an immensely large number of patients, even more when folks don't agree to the second randomization after the pre-op therapy. And you're talking about, 1,000 plus patients, 2,000 patients. If we think about the agents that we're trying to pull forward, they are probably somewhat less cytotoxic than sort of what's been used in the past. And I think the idea that we're going to be able to salvage an inferior response isn't so appealing. I think we have to think about randomizing everybody up front, biting the bullet. It makes the study about half the size. If we're doing this as a transatlantic thing, actually these trials can happen pretty quickly, as long as we can get them activated in a reasonable period of time. Finally, I would argue that that approach of looking for big differences in phase 2 trials as an approach actually worked. And it worked because other folks adopted the trial design and happened to test agents that were biologically active. And I think the class of agents that by far has demonstrated activity is really the tyrosine kinase inhibitors. These are slides, I should have put it on it, from Katie, oh I did, it's our title slide on the top left. So these are slides from Katie Janeway, and basically what it shows is you're looking for 40% or greater in the 4-month PFS. And with the exception of lenvatinib, every one of these has achieved that 40%. And so TKIs, I believe, have met the threshold. The most recent presentation has been some from the French group at ESMO and was cabozantinib. I think some of the issues are, these trials aren't meant to be compared directly to each other. So is 60% really better than 40%? Who really knows? But I think as a class, this agent has met the bar to demonstrate activity and should drive a phase 3 trial. So I do think TKI should be next. Obviously, some of the drugs like regorafenib can combine with methotrexate, so there's a lot of challenges around figuring out exactly what should be done. Again, I went through this whole long-winded thing about heterogeneity and that we have to molecularly profile. So why at the end of this am I saying we should go with a TKI? I think TKIs, particularly these less targeted TKIs, are probably not as targeted therapy as people would like to think they are. They actually inhibit a lot of different proteins. And I actually think you're back to more of a cytotoxic approach. So it contradicts that side of it because it sounds like a targeted agent that'll probably fool everybody. But at the same time, I think the reason it isn't having heterogeneity is because it's not really one drug. Again, I think it's also important not to forget what we've learned. We've consistently had these tantalizing signals of immune activity. This is one of our own papers on T-cell infiltration. We already know that immune checkpoint inhibitors as single agents probably aren't going to be effective in this disease without a lot of mutational burden. But at the same time, I think we are exploring approaches like radiation or other therapies to make cold tumors hot. Maybe that's something that we have to pursue further and just put it back on the sort of map. And I think the key is really, we've accumulated a lot of trials and data. I do think the advocates are losing patience with us because we're not making progress. And we really have to change outcomes, and that has to be the priority. So this, I think, just summarizes everything I said. I don't think cytotoxic chemotherapy is going to help. I do think genomic profiling is part of the future, both for understanding the disease as well as selecting therapies. I think we have to keep our trials streamlined. I do think we can do a TKI trial. And I think we just have to understand more of what we're doing because we're still sort of taking an empiric approach. And so from there, there's just a lot of people who contributed to all of this work, particularly within the Children's Oncology Group and elsewhere. And thank you guys for listening to this.